Everyone. So today we're going to look at gene linkage and crossover frequency. Now, when it comes to gene linkage, we're going to start with that. I'm going to just give you the title for all of it. Gene linkage is a deviation from Mendel's law of independent assortment. According to Mendel, what he said is when you have blonde hair and blue eyes, those two genes are going to assort independently. When they line up in the middle of the chromosomes, they're going to assort independently from one another. Well, Thomas Morgan, if you remember from his work with fruit flies, was the one to figure out that sometimes what you get is genes that are linked to one another. They are so close to one another that they tend to be transmitted together. So again, we're going to put Morgan and fruit fly. So remember, we looked at Morgan for multiple alleles with the fruit flies and eye color. We also saw Morgan again when we did sex linked. And now we have these gene linkage sort of concept that Morgan also brought to light. Okay, so what Morgan did is he made sort of a test cross when it came to fruit flies. And for that, I'm going to show you a picture in order for me to explain it. And I'm going to attach that. And so you can see here that Morgan had two parents. It had a fruit fly that had a gray body with long wings, which is the wild type, the most common. And you can see here that it has a genotype of G, big G, little g, big L, little l. So it is a dihybrid individual, a female. And he crossed it with a black body, short wing individual of genotype little g, little g, little l, little l. So this is another thing that we also cross. Remember, we call this a test cross, right? Now, if we were to do this particular cross where we take big G, big L, little l, and we cross it with little g, little l, what we would get is we would get a Punnett rectangle, right? So we would have big G, big L, big G, little g, big L, little g, little l from this parent right here. And then the only thing that this particular parent can give is just a little g, little l. So here I'm going to put a little g, little l. So you would expect when you're doing a cross like this is all four possible genotypes and phenotypes. So here you would have gray body with long wings, you would have gray body with short wings, you would have black body with long wings and black body with short wings. And you would expect this to be a 1:1:1 ratio. But when Morgan kept doing this, what he saw was that 83% of the time he saw the parental phenotypes, the ones that looked just like the parents, and then only 17% of the time he saw sort of that other combination of traits. So there was something going on here, and actually it wasn't Morgan who figured it out, it was one of Morgan's students who figured it out. So in order for us to see what was going on, what they proposed is, again, I'm going to use another picture. So what the student figured out is if we're looking at meiosis, when our gametes are being formed, again, what we see here is we have the parent that has the little g, little l genotype. That's the only thing that they can produce. But when it comes to the female, what we're seeing here is we're seeing sort of this crossing over occurring where we have different kinds of genotypes that are being produced, because then by taking one of these and one of these, I would get that. One of these and one of these, I would get that. One of these and one of these, I would get that, and so on and so forth. And that's how we would get these combinations. But what they proposed is the reason why the ratios were off is because some genes are linked So what does that mean that they are linked? Okay, so they are, first of all, on the same chromosome. And also, they tend to be transmitted together. They tend to cross over together. I like to think of it like they are like BFFs. They want to stay together, and we will see how this analogy of best friends also translates to the distance between genes is going to make a difference if a gene is going to be transmitted together with another. So let's say blonde hair, blue eyes was linked, then you would never be able to have, or the majority of the time you would have people with blonde hair, blue eyes, and you wouldn't have a combination of the two where you could have brown hair, blue eyes, or you can have blonde hair, brown eyes. But that's not the case, obviously, with eye color. But if they were linked, then the majority of the time what you would see is you would see those two things together, and then only very rarely would you see the other combinations. Okay, so they're on the same chromosomes and they tend to cross over together. Okay, so we're gonna go back to blue. Okay, so what you do need to know here, and then you're not responsible for knowing all the theory behind it, but what you do need to know is that the further apart two genes are from one another, the more likely they are to end up on different chromosomes. They get separated, they cross over. One stays on one chromosome and the other one jumps to the other chromosome. Okay, so the further apart two genes are from one another, the more likely they are to end up on different chromosomes. So that means that they cross over. Okay, so that being said, then the opposite is also true. The closer together two genes are to each other, the more likely they will stay on the same chromosome. So two scenarios here: either they don't cross over, they stay, or if they do cross over, they cross over together. Okay, so they're BFFs, they want to stay with each other, right? So that's the things that you do need to know. Why do you need to know this? Also, I'm going to put a little caveat under here as well. So this also applies to genes that are close to the centromere. And why is that? We talked about the centromere. If you remember, when we draw our centromere, that's the centromere right there. And remember that we said that that's a section of DNA that is really, really tightly wound. And we do that. We have it tightly wound because that's a section of DNA that we don't want to break. So the closer that gene is to that centromere, the more stable that DNA is. It's not going to break off and end up on a different chromosome. It's going to stay put. So that thing of not crossing over or staying on the same chromosome is also valid for those genes that are really close to the centromere, because chances are that centromere won't break apart and end up on a different chromosome. Okay, so I'll give you an example of what a question would have looked like for this. So for example, this is a typical diploma question. Okay, so you're given a chromosome, and on that chromosome, you have 3 genes or 4 genes. We can say we have eye color, we have body shape. So this is for the fly. Let's say that we have wing length, And then one more. Let's go for antenna number. Okay, we might have different numbers of antennas for flies. Okay, so let's say that we have that. And then the question would have been, so here I have an example. So the question would have been, which genes are more likely to cross over? Okay, so what does that mean? Which genes are more likely to end up on different chromosomes, to get separated? Okay, and let's say that I give you some options because it would have been a multiple choice question. So I'm going to say A, eye color and body shape. B, body shape and wing length. C, I'm going to go eye color and antenna numbers. And what do I have left? The last one I think is, let me see here. I lost my spot. Body shape, D. And antenna number. Okay, so those are my 4 options. So you have to choose now which ones are going to cross over to different chromosomes. So eye color and body shape. So when I look at eye color and body shape, they're pretty close to one another. And our rule says that the closer together 2 genes are, the more likely they are to stay together. So chances are is if there was another chromosome over here and crossover occurred, chances are is if this crossover happened, both of them would end up on that chromosome and the ones on this chromosome would switch over to that chromosome. Okay, but because they're so close, chances are they would go together. Body shape and wing length. So body shape and wing length has a centromere in between. So chances are is these 2 will probably not cross over because I need to have a break right here and a break right here. Now the break here might happen, but the break here is pretty close to the centromere. So probably it would not happen. Okay. Next is eye color and antenna. So these guys, look how far away these guys are from each other. So chances are is I could have a break here and this eye color would end up there and antenna could stay. Or antenna could break and go over here and eye color would stay. So then that would mean that these guys could probably cross over quite often. Body shape and antenna. So again, we're in the same spot here. Antenna could break off. Body shape is pretty close to the centromere. So chances are it's going to stay. Okay. So the answer here is we're going to look at the ones that are the furthest apart from each other, and that's eye color and antenna. So our answer here is C. Now, to finish off this section, the one thing that also you need to know is that Morgan's student was able to devise a system to figure out what the distance between those genes is according to the crossover frequency. So let's say that you have a chromosome that has 2 different genes, and I'm going to give you an example in just a second. So let's say that those genes cross over 50% of the time. He gave those map units. And so he said if it's 50% of the time, those genes are 50 map units apart. Whereas genes that only cross over maybe 1 or 2% of the time, then those genes are 2 map units apart. So 50 versus 2 map units apart. Okay, and so that's why they don't cross over very often. Their frequency is really low. Okay, so in order for us to continue with this, then also we're going to get into map units and chromosome maps. Oops. Okay, so this system was devised. So there was a system of map units. We're going to call them MUs. Okay, just MU is fine, map units, was devised to indicate the relative distance of genes from one another on a chromosome. By looking at the crossover, I'm just going to abbreviate it CO like that, frequency. So for example, this is one of the questions that you would have gotten on a diploma. It would say, can you draw a chromosome map by looking at these crossover frequencies? So we have, remember, for every percentage of crossover frequency, we have one map unit. Okay, so I'll give you some of these and this information. So I'm going to say gene A and B cross over 6% of the time. So they have a crossover frequency of 6%. Okay, that means they end up on different chromosomes 6% of the time. Okay, gene A and C cross over 10% of the time, and gene B and C cross over 4% of the time. Okay, so the way you would do this is you start by drawing a chromosome. Okay, you could draw it like that, or you can make it a fancy one, and I'm just drawing it on the side, just like that. And then the rules for this is actually quite simple. What you want to do is you want to start with the two genes that are the furthest apart. And then move your way in. So I'm going to start with the two genes that are the furthest apart. Okay, and I look here, and the furthest apart are A and C. So I'm going to place A and C on here, and I'm going to say A and C is 10 map units apart. Then next, I'm going to move my way in. So next, I'm going to go to the highest. The next highest is 6% of the time. So I know that A and B are 6 map units apart. Now, I can't put that over here. Okay, I can't put B on this side with A. And the reason for that, they're only 4 map units apart. So this is way longer than A and C. So B can't go there. So it has to go on the inside. So I move my way inward. Okay, so we said 6% between A and B. So if this is 10, probably I would say 6 is probably there. And so that's where I would place B. Just fix that. And then if we look this, we said then this and this is 6 map units apart. And then what I'm left over with is I'm left over with this little chunk of B and C. Now, 10 minus 6, that should be 4 map units apart. And according to my question, B and C are 4 map units apart. Okay? So again, you start on the outside, the very 2 furthest ones, and then you move your way inward. All right? Now, if I were to give this answer on a diploma, the reason why up here I said that what you have is the relative distance between the genes on a chromosome is I don't know if I have to go A, B, C, or C, B, A. The chromosomes flip, right? And so both of these would have been correct. Okay, so if they said draw a chromosome map and tell me where each of those genes are, then I would say, well, it's A, and then next to A is B, and then C. Or it could be, if I flip the chromosome over, it would be C, B, and A. So both of those answers are correct. Okay, so I'm going to give you another example, give you a chance to practice on your own. So example 2. So now I have diabetes. And I'm going to give these guys numbers so it'll make it easier for you to sort of make a numerical response out of it. And I'm going to say tongue rolling, so the ability to roll your tongue. These guys are 14% crossover frequency. Or tongue rolling, which in our numerical response we said was number 3. And hemophilia gene, 7% of the time. And diabetes, which was number 1. And hemophilia, which was number 4, 21% of the time. Diabetes, number 1. And let's say eye color, these are all made up. Okay. 2, that's 19.5% of the time. So again, these are all made up. If you want a real example of genes that are actually linked to one another, all you have to do is look at somebody who has red hair. And what you see is that most of the time when you see people that have red hair, they also have fair skin and good chance that they will have freckles. And the reason for that is that the red hair gene, the lighter skin color, the lighter skin tone, and freckles are linked genes. Okay, so they tend to end up on the same chromosome, and so people tend to inherit them together. And why do we have that particular adaptation? We will learn that when we do population genetics. Why is it that we have people with red hair and fair skin? And it's more common in the northern hemisphere than it is, let's say, in the southern hemisphere, okay? Or at the equator. Anyways, so I'll give you a second to pause the video and try this question out, and then I'm going to go ahead and draw the chromosome in the meantime. And you pause the video, and what we said is we're going to start with the 2 that are the furthest apart. So the 2 that are the furthest apart are diabetes and hemophilia. Okay, so I'm putting those guys on there. Next, we said we need to move our way in. So the next ones we have are 19.5 is diabetes and eye color. So diabetes and eye color are going to be, I'm going to put eye color is number 2. So I'm going to put it right here. Okay, because again, this whole thing right here is 21 map units. This whole thing right here is 19.5 map units. Okay. And then next we have diabetes and tongue rolling. So diabetes and tongue rolling is, oh, that's probably too close. We're going to go there. So that one is, tongue rolling was 3. So that is 14 map units in there. Okay, so if I were to give this as a numerical response, what is the order of those genes on the chromosome? My answer could be 1, 3, 2, 4, or 4, 2, 3, 1. And then as a last question, another thing that I could ask as a diploma, as a multiple choice, what is the crossover frequency between eye color and hemophilia? So now I can use my chromosome map to figure out the map units or the crossover frequency between 2 other chromosomes. So here I have eye color is number 2 and hemophilia is number 4. So what we're looking for is we're looking for the crossover frequency right here. So So they want to know what is that amount, okay? What I can do for that is I have the crossover frequency between 1 and 4 is 21. And the crossover frequency between 1 and 2 is 19.5. And so the difference between those two is 1.5 map units. So what I'm looking for this number, and this number is 1.5 map units. Okay, so here are the notes for gene linkage and crossover frequencies. Again, the two things that are important out of this is not so much the theory behind it or the history behind it. What I really want you to figure out is the fact that certain genes tend to be transmitted together, and what's going to make a difference is how far apart or how close together those genes are. The closer together, the more likely they are to stay together. The further apart they are, the more likely they will be separated and end up on different chromosomes. And that's how we come up with these gene or chromosome maps for different chromosomes to give us the relative distance of genes from one another. Okay, we'll see you again for another video another time.